Hi, my name is Brandon Gallas. I'm in the Division of Imaging, Diagnostics, and Software Reliability at the Food and Drug Administration. I created this video to talk about pathologist variability and agreement from a pilot study. I want to quickly show my collaborators as they've been fundamental to this work. We have expertise with diagnostic imaging, clinical pathology, digital pathology, and computational pathology. Computational pathology includes all things image processing and artificial intelligence, so very relevant today. This project is a collaboration of volunteers. I value their perspectives and contributions. We could use a little help in this area of statistics, and that's why I'm presenting today. There's room for you. In this video, I'm going to give you an overview of the HTT project, the High Throughput Truthing Project. The pilot study that I referenced is part of that HTT project. An overview of the project can be found in manuscript form on arXiv and has been accepted for publication at the Journal of Pathology Informatics. After that, I will introduce the study materials and explore the data that we collected. Spoiler alert, the data are publicly available in an R data package on GitHub. I hope you check it out. Then I'll introduce the mean squared differences endpoint that we are exploring for the data. The mean squared differences is the main quantity in a limits of agreement analysis. I'll show you some of those results. Finally, I'll close with a summary and talk about future work. Let's start with the clinical context of this work. It's breast cancer, specifically a pathology biomarker called stromal tumor infiltrating lymphocytes, and it goes by the moniker sTILs. The clinical relevance of sTILs is that it's prognostic for survival, and it's expected to inform the patient management. The more sTILs, the better the prognosis, the better the survival, so we like sTILs. A patient with high sTILs might not need toxic chemotherapies. The microscope has recently gone digital, opening the door for artificial intelligence to evaluate the biomarker. At the FDA, we refer to such an algorithm as a software as a medical device, or SAMD for short. An algorithm can reduce the burden on the pathologist and allow the evaluation process to be more reproducible and more quantitative. The deliverables we're creating in this project really center on the reference standard data from pathologists. We are collecting data that we can use to validate an algorithm. In addition to the data, we're also developing methods to validate the algorithms. These methods need to account for the complex variability from the pathologists and the correlations in the study designs that we're creating. We're going to pursue qualification of these deliverables through a relatively new program, the Medical Device Development Tool Program at the FDA. If qualified, the data and the methods can be used in regulatory submissions without further discussion. The sponsor only needs to reference the MDDT qualification. Just so you know what kind of data we're talking about, here's a slide showing a calibration cheat sheet for the pathologists to use while making their evaluations. The right-hand panel shows several examples across the range of sTIL densities. The density is a number between 0 and 100, corresponding to the amount of area occupied by the sTILs, which are the little dots in the panel, divided by the area of the stroma. I hope you can see the segmentation in the examples. This is isolating the sTILs that are in the stroma. You might be wondering how we collect the data. We have 3 different evaluation platforms in this study, and 2 of them are digital and 1 of them is the microscope. Here's a screenshot of the CA microscope platform. The image on the right shows lots of purple ovals. These are the tumor cells, and they live in clusters. In between the clusters is the pink stroma, and in the stroma are small dark ovals and dots. Those dark ovals are the lymphocytes. Don't ask me which is which. I'm a mathematician. I'll leave that to my clinical colleagues. The green box outlines the ROI to be evaluated. The pathologist can pan and zoom to get the full context of the specimen around the ROI. On the left, we see the user interface area. There are 3 evaluations to be made. The first is to label the region of interest. The second is to evaluate how much area is tumor-associated stroma. And the third collects the TIL density. The other platforms are generally the same. Our study materials start with 64 hematoxylin and eosin stained slides. The H&E stain is what produces the pink and purple colors. We pre-select 10 regions of interest on each slide for a total of 640, and we split the work into 8 batches of 8 to break the study into bite-sized chunks. One batch takes about 30 minutes. Okay, let's start looking at the data. One of the main reasons of presenting today is to introduce and share our data. And our data package is now live on GitHub. If you aren't familiar with R, a statistical programming language, the data is also included in human-readable plain text files. The primary data is the annotation data. It's a data frame named pilot HTT. The key parameters of this data frame are shown below. They include the case ID, the reader ID, and the modality ID. These are the independent variables, if you will. The dependent variables, the data we collected, include the ROI label, the percent stroma, and the density of TILs. On the right are all the all the variables available. The most important is probably the pathologist experience. Unfortunately, some of the pathologist experience information is missing, which reminds me to say this is a pilot study. There is missing data, and some of the workflow was tweaked along the way. We did our best to document the data, and we're looking for feedback to improve. The figures in this talk are generally the same as those I presented at a pathology informatics conference. Here, I want to point out that the GitHub repository has an R Markdown file that shows all the data processing done to create the figures. At the FDA, we strive to create reproducible and transparent science. For example, the R Markdown file shows the code to create the figure on the right. This figure shows the data collected with the CA microscope digital platform. The rows correspond to the readers, and the columns correspond to the slides and ROIs. Each tick mark corresponds to an ROI, which is why the axis runs from 1 to 640. 18 readers used CA microscope to collect almost 5,000 observations. The figure quickly shows us that we hit our target of at least 5 readers per ROI. And across all 3 platforms, we have collected 7,200 observations from 35 pathologists. Here's what our data looks like. This figure shows the CV, or coefficient of variation, for every ROI with data. Each circle corresponds to 1 ROI, and the mean and CV are based on averages over the readers. The horizontal lines here average over blocks of 10% of the data, or 57 ROIs. The vertical lines correspond to clinical thresholds, creating bins of low, median, and high sTIL density values. Remember, the sTIL density is prognostic for survival. The higher the density, the better the prognosis. What can we say about these results? For one, it appears that it is difficult for pathologists to quantitate sTIL density, especially below 10. A coefficient of variance larger than 1 is quite high, and 0.5 isn't great either. This plot also shows that the standard deviation is not proportional to the mean, or the horizontal lines would be at the same value. Not shown is a plot of the variance as a function of the mean. The results there show the variance is increasing with the mean as well. One consequence of these last 2 observations is that standard log or square root transforms of the data probably won't simplify our analyses. Therefore, in the work that follows, we will partition the data into 3 groups for analysis. We will also separately consider the data from all the readers and the data from a panel of 4. Think of this panel as a panel of experts. The panel of experts were chosen from pathologists that read all the data and had low levels of the mean squared difference. Since it guided our selection of the expert panel, I should probably define the mean squared difference. It's a quantitative agreement endpoint. The square root of the mean squared difference is the main component of a generalized version of the Bland-Altman's limits of agreement. Perhaps you have heard about this analysis framework. Smaller differences indicate better agreement. And ultimately, we want to evaluate the agreement between the algorithm and the pathologists. Here we see an expected value over the differences. The y values are scores from the algorithm, and the x values come from pathologists. The k and l subscripts correspond to the case and location, or the case and the region of interest. Now, a baseline level for the algorithm to pathologist agreement is pathologist to pathologist agreement. And we have a similar expression here. The j and j prime subscripts indicate we have different pathologists. Okay? I want to take one more moment to clarify the relationship between the mean squared difference and the limits of agreement. With some basic algebra, we can see that the pathologist to pathologist mean squared difference is really a variance. And the square root of this variance is the standard deviation that goes in the limits of agreement expression. So here's what we found. There's a column for the results for all the readers and one for the panel of experts. The different rows correspond to the low, medium, and high sTIL values. I want to make sure you know that these limits of agreement account for both reader and case variability. They also account for the correlations arising from the data collection study design. We often refer to this variability as multi-reader, multi-case analysis, or MRMC for short. Of course, it's interesting or nice to see that the variability decreases for the panel compared to all the readers. We did, after all, choose the individuals that had good agreement, so that's not a surprise. In terms of the levels of sTIL density, we see that the variability increases from low to medium to high. And we expected this trend given earlier observations. I want to point you to some written work that these results are based on. C. Wen's work uses an ANOVA with reader and case random effects. The manuscript also includes a simulation model for validating the method. Complementing this work is a GitHub repository which, like the work described here, includes functions and scripts that can replicate all the results in the manuscript. Before moving from this slide, I want to point out two things on our to-do list. The ROIs are nested inside the images, creating a potential for correlation. We still need to account for this correlation. Additionally, we need to estimate the precision of the limits of agreement estimates. We need confidence intervals for these results, and we're working on these methods. So that's my talk. We provided an overview of the project starting with its purpose, which is to create a data set of pathologist annotations to validate an algorithm that estimates a biomarker, the sTIL density. The first step is to understand pathologist agreement, or pathologist variability. I showed you the methods, platforms, and materials to collect the data from pathologists and showed you what the data looks like. This is not independent and identically distributed data. It will take sophisticated methods to treat the correlations and complicated non-stationary behavior observed. We still need to account for correlations between ROIs in an image and estimate the precision of the limits of agreement estimates. We are also exploring different analysis methods, non-parametric regression, rank-based correlation, and categorical agreement. Finally, and most importantly and urgently, we need to size our pivotal study. So I hope I've intrigued you with the purpose of this project and the challenges we have to analyze the data. Please check out the data on GitHub, and I invite feedback and questions via the issues page or reach out to me by email. Thank you for listening today.